I think Devin sent out an email. Did all of you get it? There's no lab this week. I think so. And the last lab is next week, and it's going to be doing all of these predator-prey isocyclines and competition isocyclines. And we're going to finish up the predator-prey interactions today, and then we're going to move on to sort of interspecific competition. I'm just going to give you a little warning. This is sometimes excruciatingly painful for students, okay? And again, what we're going to be developing are what are called isocyclines. And these are basically, and again, you don't have to understand how to solve differential equations and isocyclines and all those things. I'm not going to ask you to determine all these equilibrium points, maybe some of them. I'm going to ask you to really, what I really want you to understand are these graphs that I create that are these competition isocyclines and these predator-prey isocyclines. And you'll be doing that in lab quite a bit, and that'll be before the exam, so I think it'll be very helpful. So, and what I'll do, I don't think I'm not going to jump into the competition isocyclines and the derivation of the competition isocyclines until Tuesday. So, but I'm going to try to set the stage for interspecific competition and under what conditions one species can exclude another species and how they might coexist. And we'll talk about some examples of that. Okay. I'm going to go through the predator-prey examples today, finish up the predator-prey, and then and we'll do a little extra credit question in the middle of it, and then we'll launch into competition. And then at the end, I'll ask you to turn in your homework, and I'll go through the solutions to that. Okay? So. So one of the key, so let's finish up the predator-prey stuff, and I'm going to be developing these predator-prey isocyclines. And one of the things, the key theme that we were talking about is what are we seeing in terms of the population numbers of predator-prey? What happens in a lot of situations, whether it's this one or the snowshoe hare lynx example? What's the main thing? Yeah, it correlates and they oscillate. They oscillate from high numbers to low numbers, and often the predator numbers are slightly behind the phase of the prey numbers, right? So predators eventually get to high numbers, but it's lagged in terms of the peak abundance relative to the prey. So what we see are not stable numbers over time, but these sort of oscillations and these time-lagged oscillations. One of the things I want to try to address today, we're going to do the predator-prey isocyclines and show you how you can get these oscillations, but one of the things I'm going to focus on at the end is, again, one of the themes I talked about at the beginning was, what's driving those oscillations? Is the predator from the top down driving it, or are the plant population, are the plants driving it? And so we'll talk at an example at the end about the snowshoe hare and lynx and how the plants might be driving it, and then I'm going to ask you a simple question. Okay. So we're really interested in these sorts of population dynamics that lead to oscillations. And again, here was an example where maybe a predator is kind of driving it, but this is the case where there's no predator in it and it's still getting these oscillations. So we're going to try to figure out what's going on here. Okay. And they have dramatic effects. Here we go. So you're going to be doing the lab next week. You're going to be looking at what are called predator-prey isocyclines. This was a group of Russian scientists, and this was about the 1918, 1920, they developed this model. And it was Lotka and Volterra, and they developed a model of predator-prey interactions. And we're going to define very simply, prey are going to be V or victim or prey, and big P is the predator. Okay. And again, what we're looking at here is what influences the change in the number of the victim or prey over time. And this is the equation for it, and we'll get to it. And then this is the differential equation to look at the change in the predator numbers over time as a function of these two. Okay. Again, I'm not going to ask you to memorize that. I'm not going to ask you to understand all those details of how these things are calculated or anything like that. But the key thing to look at here, let's look at the prey numbers. There are two components. You can see one is positive, okay, and one is negative. Same down here. You have a positive component and you have a negative component. So some things contribute to positive growth rate of a prey population. Some things have a negative influence on it. And this is the basic differential equation. Okay. What's this look like? Does this look familiar? If you were to exclude that part of the equation, what would you have? That's the small r. That's that population growth rate, right? And if you have nothing else going on, if you excluded that term, what would you get? what would the population growth rate look like? It'd increase without limit, wouldn't it? That's kind of the exponential growth, right? So as long as that's positive, that will, in the absence of this, you'll get positive population growth rate without limit. So this places a limit on the population growth rate of the prey, and what do you think it is? What's limiting the population growth rate of those prey? The predator, okay? And it's a function of both of those. Again, you don't have to worry about it. You multiply all these numbers together to kind of get this negative effect on the prey population growth rate. So V is the number of prey, P is the number of predators in the population, right? Here's the key variable right here is this C. And this is basically where the first part is the intrinsic rate of growth, and the second part, which is negative, you can think about it as the removal of prey by predators, okay? And C is a variable that can range all over the place. You can have a low capture, predators aren't very efficient or don't eat a lot of these prey, then you have a really low capture efficiency. If this is sort of a specialized predator that eats a lot of these things, then you have a really high capture efficiency. So this value can change a lot. I don't think in your lab you're messing a lot with these values to determine this, so you don't have to worry too much about this. But you can think about this, the second term is basically the removal rate of the prey by predators. And think of C as kind of the capture efficiency of a predator. And a lot of that has to do with how much they're specialized, how good they are at eating those prey, capturing those prey. There are lots of things that can influence that. Okay. So this is, and we're going to solve for this equation to determine these isoclines. So we're going to come back to this. Here is the predator numbers. Okay. So let's deal first. So this is the change in the predator numbers over time. There's a negative component and there's a positive component. Okay. Let's look at the negative component first. Minus D is basically death rate is usually some sort of constant. And it's not due necessarily to starvation. It could be, but it's just some sort of constant death rate that occurs in the population multiplied by the number of predators. So there's some sort of characteristic death rate of predators over time that reduces the population size. Okay. The positive component, again, is a function of both the predator numbers and the prey numbers. And A and C, you've seen C is kind of the capture efficiency. So that obviously if a predator is efficient at capturing prey, that's going to translate into more predator offspring and population growth, right? The other component here, and you don't have to worry too much about it, is basically this first part is the birth rate, the number of prey captured up in here, C times VP, multiplied by A, which is basically the efficiency with which food is converted into predator offspring. Okay, and again, there are lots of things that influence this, multiple things that influence this. But a lot of times when you think about that, when you think about a predator, if it eats a bunch of, if a lynx eats a bunch of snowshoe hares, does it immediately convert that energy into offspring being born? It takes a while, doesn't it? There's a gestation period. They have internal fertilization. The babies grow inside mom, right? Develop inside mom, and it takes often months, maybe 3 months to convert, once a female is mated, to offspring being produced. So it takes a while to convert that increased food availability into offspring and into population size. So this efficiency with which food is converted into, and a lot of times when you eat food, right, you lose a lot of energy, right? So obviously you excrete stuff, you respire stuff, all these things that result in a lot of energy being lost and can't be used in offspring production. Okay. So these are the two equations. Again, you don't have to worry too much about it, but what I do want to show you is how you derive these isoclines. And okay, this is where we're headed. Okay, but I'm gonna solve so this is kind of the key points, and then we're going to put these two things together, and we're going to show how this causes these oscillations. Okay, where am I? Okay. So here are the two equations that we had. The prey isocline is up there, RV minus CVP, and the predator isocline is down below. So do you kind of remember the moose example where we developed those lines that if you're above it, something increases, if you're below it, something decreases? These are called equilibrium lines. We use this a lot in sort of mathematical modeling in biology, but what we're going to be developing are what are called lines of equilibrium, okay? And the first one we're going to develop is what's called the prey. So these lines of equilibrium are called isoclines, and they're derived from that equation. And the way to do that is you basically just, again, you don't have to worry about it. You don't have to do this. You're never going to have to solve for these equilibrium points. But you solve for a differential equation and equilibrium line by setting that value equal to 0, basically saying the change in population size over time is not changing. It's reached an equilibrium, okay? So you do that by setting each one of these equations equal to 0 and then solving for it. And what answer do you get? I haven't had algebra in 35 years. Okay, it's pretty simple. All you do is you set that equal to 0 and you say, whoops, maybe I should, RV equals CVP, okay? Now obviously you have two variables are on the same side, right? What can you do with V? You can eliminate that, right? So you can just eliminate those, and what you have, R equals CP. And what you're solving for in all these equations is the big P or the big V, okay? So the equilibrium point is P equals, so what's that value? R over C, okay? So that is the equilibrium point for the Prey isocline. Okay. Now what I've done is, here's the graph over in here, and I plotted both of the isoclines. So what we've solved for is this first one, and you'll think, well, why are you putting it there? I'll try to explain. Okay. This is the Prey isocline. This is the predator isocline, and we'll eventually get to that. Okay? So all that, and you'll see this on the graphs that are coming, this equilibrium point on this axis right here is, whoops, you want these color coordinated, right? That is R over C. That's that point right there on that axis. Okay. Let's look at this graph and say, this is, you can see this is the Prey isocline right here. Okay. Let me ask you a question. Below the line, the population of prey is either going to increase or decrease. Above that line, the prey population size is going to increase or decrease. Which way do you think it's going to go? Take a shot. What do you think is going to happen below the line? It's going to decrease? Damn. Darn. Anybody else want to go for the alternative? It's going to increase. Okay. The way, and this is conceptually kind of difficult to understand, believe me. Think of this line. You got to think about this line here. It tells you about the relative number of predators that they're experiencing, right? This is lower, right? That's sort of higher. Think of that scale. Now what do you want to do? Now do you get it? So below this line, what's happening is, what's going to happen to the predator, the prey numbers? They're experiencing, they're below that isocline, that equilibrium line. Let's just think of it as there are relatively fewer numbers of predators in the environment. So what should happen to prey population size? Yeah, it's going to go up. Now again, here's the other, so you can put an arrow in here, and remember, prey is along this axis, is along the x-axis. Which way do you want the arrow to go? To the right or up? To the right. So what that's saying is, when you're in that kind of quadrant, the prey numbers are going to go up because they're experiencing relatively few numbers of predators. And it's also true over here, too, isn't it? What about up here in those two cells? And we're going to come back to this, and we're going to show you how to. What about up here? Which way should the blue arrows be going there? What kind of environment are they in, relatively speaking, about the number of predators? A lot of them? Yeah. So where do you want it to go? Yeah. Okay, so keep that in mind, okay? And this is the key thing I want you to be able to understand is, when you're below an isocline, what happens, and when you're above an isocline, what happens? Okay. Now let's do the predator. How do we solve for that? Same thing, right? Whoop. What color are we? Red. Okay. ACVP equals DP. Okay. What thing can we get rid of? The P's. Solve for what? V or pray, right? V equals what? Yep. D over AC. Okay? Where do I put that equilibrium point? You want to put it right here? Yep, that's where we want to put that point. Again, you're not going to have to memorize that or do anything like that, okay? But, okay, now let's see, that's the predator isocline. See, the predator isocline intersects the prey axis, right? So what kind of environment is in here, in this area? What's the relative availability of prey to that predator? Is it kind of low? It's below the isocline. So what do you think should happen to predator numbers? Which way should the arrow go? Yeah. You want it to go down? Yep. What about up in the upper left-hand quadrant? Which way should the arrows go? Just think of where you are. Remember, this is low prey availability, high prey availability. Still down, right? Okay. Now, what do you want to do when you're above that predator isocline? What kind of environment are they in? Lots of prey? Which way should the arrows go there? Yep. They're going to go up. And in here, they're going to go up. Okay? So we're going to go through each one of these quadrants in another graph. So let's go back to the PowerPoint. Okay? And this is what I kind of just showed you, but I had put the other lines together. Okay? So let's look at this. So again, here's the prey isocline. That's that equilibrium point that we derived by setting the differential equation equal to 0. There's the other equilibrium point when we set the predator population size over time equal to 0. This is the predator isocline. Here's the prey isocline. So the key is prey populations increase in this region of the graph, right? Because they have fewer predators. You're below that isocline. What happens up here? Prey populations decrease in this region of the graph. Remember, this is the number of prey on the x-axis. Prey populations decrease in this region of the graph because they have many predators. Lots of predators up in here, low predators down in here. Now, the predator populations decrease in this region of the graph. The arrow's going down. Remember, number of predators is on the y-axis. The red arrow is going down. Decrease in this region because there are too few prey to eat. This is relatively low prey numbers in the environment. Up in here, above that isocline, you have predators in this population increasing in this region of the grass because they have many prey to eat. Okay? Is that, you look perplexed. Are you perplexed? Okay, let's move forward. Now we put these two together. And what we're eventually getting to is you're going to get oscillations. Okay. Now I'm going to go through each one of these. Go back to that graph that I did where I put the arrows in, right, on the iPad. Here's the key point here is there is only one point where the population sizes of both the predator and prey do not change, where there's what's called a joint equilibrium, and it's right at that intersection. That's where the change in both the prey population and the change in the predator population are where those two lines cross. They're actually equal to 0, right? And they will not change. You'll do this in lab. What we're going to have you do is a bunch of simulations where you try to get the point exactly on the starting population size, exactly on that equilibrium. And hopefully what you'll see over time is nothing ever changes. It's a stable equilibrium. No, this is just purely mathematical. So this never occurs in nature. Okay. But what we're really interested in is what happens when you move. That is a stable equilibrium, but once you move away from it, it causes instability. And that's what we're going to go through here. Okay. So let's start. Think of this as quadrant 1, quadrant 2, quadrant 3, quadrant 4. What you will get is basically what does this represent here? It represents a circle, and it represents the changes in the population size of both the predator and prey over time. And this you can think of as kind of one cycle where they go from low abundance to high and back down to low. That would be kind of considered one cycle. And what this model does is it generates these oscillations, and I'll show you why. So let's start here. Imagine, and you're going to do this in lab, you're going to start where you put a point here where in one of these quadrants, and you're going to watch to see what happens to the population sizes over time. Let's start in this quadrant. What's happening in that quadrant to the relative numbers of predator and prey? Look at this arrow. What's happening? So think, for the predator, you got lots of prey. What's happening to the population size of predators? It's going up, okay? You're also in a quadrant, relatively speaking, where the number of predators is what? Yeah, it's relatively low. So what's happening to prey abundance in this quadrant? So this is the quadrant where both of them are increasing, okay? They're kind of in the parts of the ice, above or below the isocline where they're both going to be increasing. So what this arrow represents is the joint population sizes of both the predator and the prey. Prey are going up, right? And predators are going up. Okay, so this is positive population growth rate for both. Okay? So that's what happens entirely in that quadrant. What happens when you cross one of those isoclines? Did something change? Yep. So this is good. Now you're above the predator isocline, right? What's happening to prey numbers? What kind of environment are they experiencing? A lot of predation, right? So what you can see is this now has started to go in this direction. So in this quadrant, the number of prey are decreasing due to high attack rates because of high numbers of predators, okay? What about predator numbers? Yeah. So what you can see is they're still in the quadrant where they're relatively speaking, this is a little counterintuitive, they're still in an environment where there are lots of prey out there, right? So they're attacking them at a high rate, and their population size, what's happening to the predator population size in this quadrant? It's increasing. Okay. So plus, this is minus prey plus predator. Now you cross the predator isocline. Now what's happening to both populations? They're both decreasing. Okay. So here, the predators now are in an environment where there's low prey availability. Their numbers are declining. What's happening in terms of the number of predators? They're still above their isocline, high attack rate. So both the predator and prey in this quadrant are going down. So there's a negative growth rate in that quadrant for both. Then you cross this final isocline. What do you get? Yep. So here, now you have relatively fewer numbers of predators, right? because they've died for various reasons. What's happening to the prey numbers? They're increasing. And then you're still, think about here, you're below that prey isocycline. What's happening to the predator numbers? They're decreasing. So here, so these are kind of the situations on this diagonal where one is increasing or decreasing and vice versa. So these are kind of where both are increasing or both are decreasing, right? So here what you have is this is the case where prey are increasing and predators are declining. And eventually you get back to the point where it reaches the start again of this entire cycle. Okay. So what I'm really interested in is that you kind of understand the dynamics of this, and let's show you, how does this translate into the joint population growth? Yep. The first quadrant, what's happening here? Both the prey and the predator are increasing in population size. So those have positive population growth rates for both predator and prey. And so the key, you kind of have to kind of look at the equilibrium line and then these axes in terms of, so, okay. So let's then translate that into a graph. And this is what you're going to be doing in lab. You're going to be doing this exact same thing. So basically what we're doing is, let's start right here. This line right here. So what we're doing is we're following the joint effects, the joint numbers of predator and prey over time as we go through each one of these quadrants. Okay, I'm not going to go through all of them. I'll just go through a few of them. Okay, and you might, as an exercise, start going through these. So let's start right here, where we're starting basically right in there. Okay, what's happening? They're both increasing. Okay, what's that blue line right there represent? Pretty much when you cross over into that quadrant. So we're going from first to second to third to fourth. And what we're translating that into is the joint population dynamics of the predator and prey as this, okay? So now this line represents where you cross over into this quadrant. What's happening to the prey population size? It's decreasing, but the predators are increasing, even though they're prey. And a lot of this has to do with the fact it takes a while. Predators have a lot of prey to eat, and it takes a while to convert those offspring, that increased food into offspring. So that's what creates this lag, so that the predator abundance is lagging behind the prey abundance, especially for organisms, a predator that has a longer generation time or just longer gestation period, takes longer to convert food into offspring. Okay. So there you've got, should I just go through all of them? Why not? Okay. So here we got this. Now the predators are still increasing, the prey are declining. Now we're crossing over into that quadrant. Now what's happening? Yeah. So here now we're crossing over into here. Now the predators are declining and the prey are still declining. Okay. And then finally, when we cross over into this point, I don't know why they can't align these very well, but they don't. So anyway, this is where we cross over into this. And what happens here? So when we cross here, what do we got? What's happening to prey numbers? They're increasing while the predator numbers are still declining. Okay? And so what this represents is this actually represents one entire cycle. Okay? And again, is this kind of what we see in the snowshoe hare lynx example? So this mathematical model kind of mirrors or mimics what we often see in predator-prey dynamics, where you have these coupled oscillations in the population dynamics of both predator and prey, and often the predator peaks and decreases are offset from the peak abundance of peak low numbers of prey. Okay. So there's kind of, as the prey population falls right in this quadrant, it will support fewer predators, so they obviously start to decline, blah, blah, blah. Okay. So those are kind of the explanations. So let's kind of go, so what I really want you to understand is what's happening in each one of those quadrants, and I think that the computer simulations you do in lab will help you understand that pretty well. Okay. And in some sense, once you get outside of that point where the two lines cross, you get these oscillations, and you don't have a stable equilibrium. It kind of goes through these oscillations. Okay. So let's kind of, again, what we see. This is kind of an example over years. This is a more recent study from the Snowshoe Hare, not from Hudson Bay, but people in Canada are still studying this. And here's the number of hares per 100 hectares and the number of lynx per square kilometers. And what you can see is you see an increase in snowshoe hares. Lynx takes a while. It's offset a little bit, right? Their peak abundance is a little later, okay? And then you see the decline, okay? And the cycle usually runs basically, this is 1, 2, 3, 4, 5, 6, 7. It's a 7 to 8 year cycle to go from low abundance to the next low abundance. So it's about an 8 year cycle, okay? What are predators doing this time? Are they just, what does this show? So when snowshoe hares start going to fairly low abundance, right? Especially in that's when they're kind of getting low, 92 and 93. What are lynxes doing? So you can see that these are kind of the number of attack rates by the lynx predator. These are little small mice. These are squirrels, snowshoe hares. What do you think is the most profitable prey out of those three, if you had to guess? Snowshoe hares. They're much bigger. And in terms of actually their search time, how long it takes to find them is not that much more. Snowshoe hares are definitely the preferred prey item of lynxes, right? But what happens when their profitable prey item gets at really low abundance? What do? They switch, right? Okay. So they'll switch to other things to eat, and they'll switch to red squirrels or these are little small mice, right? And they're not so good. Does it translate into much? They're still declining, okay? So they're kind of do as much as they can with an alternative prey item that probably isn't terribly profitable. And they're going down as the snowshoe hare lynx goes down. Okay. So they can switch. So here, I'm about ready to put an extra credit. So let's look at this. So this is a little different presentation, but lynxes are along this axis and hares are along this axis. And you can kind of think of this as a circle, right? Leading to a cycle, basically from 87 to 94. So about a 7 to 8 year cycle. And here's what's happening to the relative population sizes. Hares are at 100 hectares, right? Then they're increasing up to about 200, right? Between 1987 and 1988. Then what happens? They're declining. What's happening to the lynxes? They're increasing, right? So again, here's the phase where you see the hares and lynxes increasing, that bottom right-hand quadrant. Now they're declining. One of them is declining, the hares, and the lynx are still going up. And then now you're crossing into the third cycle where they're both declining, right? And then you're eventually getting to the point where the prey increase and both of them are starting to go up a little bit, okay? So the question is, what do you think happens to the food quality for hares when they are at these really high densities? What do hares eat? Grasses, shrubs, all sorts, usually low-lying vegetation, right? Those sorts of things. So there are two alternative hypotheses. There are kind of two hypotheses that could explain the drivers of these oscillations. One is the food resource availability, right? So when snowshoe hares get to really high abundance, right, what's happening to the food quality? It's going down. It could be for a lot of reasons. They're overgrazing it. The plants could also be sort of producing more secondary compounds and toxins to kind of defend against it. Turns out that's probably not the main thing, but some of them seem to do that, okay? So the question is, what's causing these oscillations? Is it primarily, are the lynx the primary drivers of it, or is it kind of, so this is sort of would be what's called top-down control, right? The predators are driving the lynxes through these oscillations. by causing high mortality, then they eventually fall. The other alternative is the plants could be driving this. And the lynxes just kind of follow along, right? So the other alternative is, as these hares increase in quality, increase in numbers, right, they decrease their food availability, and they die. And what happens to lynxes? They die, right? And so the question is, what's really the primary driver of these oscillations? So do you want a little extra credit question? I want you to go into Canada and design a simple field experiment, actually in the field, where snowshoe hares and lynxes occur, right, or in the plants. Design a simple experiment to determine whether lynx are the primary drivers of oscillation and hare abundance, or whether plant resources are the primary drivers of oscillation. And what I want you to do is I want you to just quickly, briefly tell me what would be an experimental treatment that you would design to distinguish between those two. Now, I know this is really hard, but do you want a hint? Nah, I'm not going to give you a hint. Okay, so I want you, there's actually a really simple experiment that you could, if you could do something and still get oscillations, it would implicate one being more important than the other. Okay, okay. I'll give you 5 minutes on this one. 5 to 7. Don't do the demography homework while during this time. Okay. Okay. How are you coming along on this one? Ready? No? Almost? Okay. 2 to 3 more minutes. Okay, ready? Yeah, ready as you'll ever be. It doesn't matter what you write. You always get a point, right? Yeah, it is. I shouldn't say that. I don't even, yeah, you're exactly right. I shouldn't say that. Thanks. Anybody else? We'll do a quick eye clicker now, and I'll talk about what I was kind of looking for. You can eye click in for attendance at any time. Oh, it'll stay up. Is that it? Everybody turned in here? Okay. So what'd you come up with? What's a simple experiment? Okay. Yeah. So that would be a good experiment to provide additional food for the hares, right? So that they're not food limited, and see if you still get the oscillations. Okay, that's a good one. Anybody come up with anything else?
I took out the prey, and then I also limited the amount of food.
Okay.
The hares.
Okay. I probably wouldn't remove that.
I was trying to see if food helped.
Okay. I probably wouldn't do that. I was thinking of another removal experiment. Yeah, so this is what people have done. The best experiment to do is to actually, they've set up these big exclosures, where guess what they're excluding? The lynxes, okay? And so there are these long-term experiments going on where they actually set up these huge exclosures where they're only the snowshoe hares and the food. And so they've removed the predator from the dynamic, okay? So these are what are called exclusion experiments. These are what ecologists do all the time. So one of the things you can do is you can remove one level of the one of the participants from the interaction and see what happens. And so what result would be consistent with food driving the interaction in the absence of predators that you what would the outcome be that would support that hypothesis? Yeah, that you still get those oscillations, right? Okay. So anyway, those are good. Anybody come up with anything else? Or you don't want to tell me what you came up with? Okay. Okay. So that's kind of what I was thinking. Removal experiments are often removing the sort of the top level driver is what most people tend to do. Okay. Let me just say, go ahead and you can I-click in. I got to save this recording if you haven't done it yet.